right now.
Right, let me share my screen.
Is anything stopping you?
No, just making sure I have everything. My computer is still recovering from all of the R21 submission from yesterday, so. All right. Are you able to see my screen?
Yes.
Okay.
I can see
All
your
right.
full PowerPoint window. It's not in presenter mode for me. There we go. It is
I
now.
can, yeah, am I audible?
Yes.
Okay. All right. So, good morning, everybody. I'm Sriganesh Rao. I'm in the Buffalo. My lab wears several hats. My initial training was with Dr. Steve Fleisler, and my prime scientific interest is actually in cholesterol homeostasis of the retina. But as we all know, lipid homeostasis is not just about synthesis, but it's also about uptake and efflux. So if we are really to understand lipid homeostasis in the retina, one of the first things we need to understand is how endocytosis works in the retina, and the specific cell type of interest for me is actually the photoreceptors. So I had this parallel interest during my initial training phase about endocytosis mechanisms in photoreceptors, and unfortunately, there is not much known about endocytosis. And also, during my initial training with Steve, one of the important problems I was studying was um, the this yet to be fully understood connection between cholesterol and autophagy and how it may be affected in the retina. So I slowly started moving towards studying endolysosomal biology and autophagy in the retina. And I'm going to be presenting some of the progress we have made over the last 2 to 3 years on this problem. So this is the title of my talk, Photoreceptor lysosomes, where do they all come from? Where do they all belong? And I added another statement, what do they all eat? Yeah. Why is it important to understand lysosomal biology? This is really biologically significant because it provides us a foundational understanding of how organelles are distributed and trafficked in photoreceptors. And we are also trying to identify the site of endocytosis in photoreceptors. This is actually very challenging in neuronal systems to study where exactly endocytosis is occurring. There is some preliminary evidence that endocytosis occurs in the axonal initial segment, but when it comes to neurons in the CNS, the cell axon is about 20,000 times longer than the size of the soma. So studying endocytosis in neurons is extremely difficult, whereas in case of photoreceptors, we have this really beautiful organized structure which we can leverage to actually study endocytosis in neurons and using photoreceptor as a model system. People alternatively culture neurons on microfluid chips and image organelle trafficking using these chips. But in case of photoreceptors, we have these neurons really nicely combed, so we can actually study organelle trafficking in photoreceptors. And why is this important? Because some of the evidence that I'm going to show today um, is going to clearly demonstrate how these processes of endocytosis, lysosome biogenesis, organelle trafficking is significantly affected in some common inherited retinal disorders, specifically in CRUMS1, and it also has direct implication in the pathogenesis of diabetic retinopathy and AMD. So there are some shared pathological features between these varied retinal dystrophies, retinal disorders. It also has an implication in some of the drugs that's used for cancer therapy, such as zinc alkaloids, such as vinblastine and vincristin. And we also demonstrate some bottlenecks, specifically in CRB1 targeted gene therapy. All right. Why photoreceptors? Photoreceptors is functionally and structurally stratified. We have the modified primary cilia, which is the outer segment. But as we all know, the real estate in the outer segment is just filled with the disc membrane, and there is no real organelle presence per se in the outer segment. However, because of the tight packing of photoreceptors in the retina, most of the organelles are jam-packed in the inner segment, and within the inner segment also, there is stratification where mitochondria is enriched in the ellipsoid, and the ER Golgi complex is in the myoid of the photoreceptors. And right underneath the myoid is where the homotypic and heterotypic adherence junctions are, where photoreceptor adherence junction, and photoreceptor glial adherence junctions are formed. And then we obviously have the cell body or the outer nuclear layer, and we have the photoreceptor presynapse. So the goal of this study is to understand how exactly different organelles are distributed in this stratified neuron. Now, neuron, retina, and RPE cells are post-mitotic. So understanding autophagy and lysosomal biology in this post-mitotic cell is extremely important. Now, phagosomes have been documented to occur in the myoid region by some of the early works in 1990s by Reme and others. As far as endocytosis, this is a mythical creature. How do you catch an endosome even in the photoreceptor? However, back in the 1980s, Joe Holyfield did this really cool immunogold electron microscopy study where they clearly demonstrated uptake of IRBP in the inner segment. So you see the immunogold positive label and sitting within an endosome in the inner segment. So there is some preliminary evidence for endocytosis occurring in the inner segment myoid region. Okay, and there are some other indirect evidences. I'm just showing you some. The work by Jeannie Chen's group shows enrichment of adapter protein 2 required for endocytosis in the inner segment, also that of clathrin. It's kind of hard to see here. As far as late endosomes, Dr. Ted Wenzel's group showed that knocking out VPS34. Let me actually try to zoom in on this image here. Bam, right there. So if you look at late endosomes, it appears in the myoid and then it appears in the photoreceptor cell body. But however, in their VPS34 knockout, it becomes very obvious that these late endosomes are in the myoid, the photoreceptors, and it's in the cell body. We independently actually verified this. We published a paper in 2021, 2022 on how to monitor autophagy in the retina. So let me just give a quick background as to how exactly this tandem tag strategy works. Back in the late 2000s, the first autophagy reporter mouse model was generated where EGFP was essentially latched on to a phagosomal membrane protein LC3B, and you essentially have green fluorescent phagosome. But however, the pKa of GFP is about 6.5. So when a phagosome fuses with the lysosome, the GFP fluorescence is quenched. So the mature phagolysosome actually does not have any signal. So if you only use EGFP LC3, we can only capture immature phagosomes, but you cannot capture mature phagosomes. Fast forward 6 years, in 2014, Joe Hill's group at UTSW developed a tandem tag autophagy reporter mouse where LC3B had a tandem MRFP EGFP tag, wherein the pKa of MRFP is about 4.5. So it's resistant to the lysosomal pH, and it does not get quenched. So we essentially have this dual signal system where immature phagosomes that are yet to fuse with lysosomes are positive for both RFP and EGFP. But upon fusion, you lose out on the EGFP fluorescence and you essentially just have the RFP fluorescence. So when we looked at the retina in this mouse model, it was very clear that the phagosomes and the lysosomes appear in the myoid, followed by its appearance in the cell body. So we got really interested in what exactly is determining this distribution of lysosomes in the retina. So here is just a C57 Black 6J retina. We have labeled it with phalloidin in red and lysosomes in green. And again, you can very clearly see lysosomal enrichment in the myoid, very few lysosomes in the ellipsoid, and they also seem associated with the actin cytoskeleton. And then we see this beautiful distribution of lysosome all across the cell body slash ONL. So there are a few facts that we need to appreciate here. One is that the actin cytoskeleton is obviously enriched in the inner segment, and it's essentially serving as the photoreceptor cell cortex. And this is one of the facts that I teach my students in the BMS 501 cell biology class here at UB. The mechanisms of endocytosis. And actin cytoskeleton is indispensable for endocytosis and endolysosome genesis. That is because actin cytoskeleton is acting as force generating machinery to pull the endocytic vesicles in. So if you don't have actin, you cannot pull the endosomal vesicles in from the plasma membrane. And what happens after the endosome is internalized in the cell is that the clathrin coat or the caveolae coat is removed. And now the naked endosome, the naked newly formed endosome, sits on a microtubule and it's trafficked on the microtubule.
If I may,
So,
I'm sorry for
yeah,
interrupting. Are you okay with interruptions?
yes, of course, please.
Okay.
Anybody, if you have any questions at any point, please feel free to interrupt
Okay.
me.
How do you distinguish between Mueller cell lamp 2 and photoreceptor lamp 2?
Good question. You can. So some of the lysosomes that we are seeing here could also be in the Mueller apical microvilli and also the Mueller cell body. That's a point well taken. But of course, there are now ways of being able to visualize and investigate lysosomes in a cell type specific manner. There is a lysotag mouse which can be conditionally knocked in the cell type of interest. So yes, that is actually one of the things that we would like to do in the near future. But based on these facts, what we hypothesized as to what's describing this distribution is that endocytosis and endolysosome biogenesis may be initially occurring in the inner segment in an actin-dependent manner, and the presence of the endolysosomes and lysosomes in the photoreceptor cell body is then explained by its microtubule trafficking along the photoreceptor cell body. So how do we test this? There are two parts to this hypothesis. One is that there is endocytosis is occurring in the inner segment, and then there is subsequent microtubule dependent trafficking. So are there ways we can modulate the microtubules and the actin cytoskeleton and see if the endocytosis and trafficking can be separately inhibited? Okay, so how do we develop methods to quote unquote remove actin cytoskeleton or microtubules in the photoreceptors? That's pretty challenging. But before we did that, we needed a tractable way of first being able to stratify the photoreceptor into multiple layers, that is the ellipsoid, myoid, the ONL, and the presynapse. And in order to do that, we decided to utilize MitoMK2 mouse model. So this is a MitoTracker mouse model where MK2, a red far red, it's not strictly a far red fluorophore. It bleeds in both the red and the far red channels. This MK2 fluorophore is targeted to the mitochondria by using a COX-8 mitochondrial translocation signal. So they're essentially generating the fluorophore in the cytosol, and it gets kidnapped into the mitochondria, so you can easily visualize mitochondria. And as you can clearly see, MK2 is in red. That's the mitochondria. You see this beautiful mitochondrial signal arising from the inner segment. There's really not much happening in the myoid. So that is the space between the ONL and the ellipsoid. That is the myoid. And you have the photoreceptor cell layer, the cell body, and the photoreceptor presynapse. And we can again see that lysosomes are enriched in the myoid and all across the photoreceptor cell body, and there are some lysosomes in the photoreceptor presynapse. What I would also like you all to observe is that we see these strawberries all across the photoreceptor, where, see there. So those are, we obviously cannot at this uh, resolution clearly tell that those are mitochondrial lysosome contact sites, but those organelles are opposed against each other. We have verified this even in 3D visualizations. They all clearly show a lysosome essentially sitting right on top of a mitochondria. But notice that it's really hard to see any prompt mitolysosomes where mitochondria has been targeted to a lysosome. Okay, we might be able to catch a few in the synapse, but almost no mitolysosome events, but a lot of mitochondrial lysosome contacts. But these contacts are obviously extremely important for organelle homeostasis, for calcium homeostasis, lipid homeostasis. But one of the things that we did with this mouse model was we were now able to, just using ImageJ, distinguish between ellipsoid, myoid, cell body, and the presynapse, just based on the mitochondrial distribution. And we just calculated organelle density in individual layers as percentage area occupied by organelle X or organelle Y. So what we see is that obviously for mitochondria, its highest density is in the ellipsoid, and its next highest density is in the photoreceptor presynapse. And there is some mitochondrial density in the myoid and the photoreceptor cell body. We have also measured the size of mitochondria and lysosomes, specifically in the photoreceptor cell body, and mitochondria is significantly larger than lysosomes. As far as lysosomes, their density profile is slightly different from mitochondria. As we initially had noticed, it has the highest density in the myoid and the cell body, followed by the presynapse, followed by ellipsoid. So lysosomes are enriched in the myoid ONL, and it probably gets trafficked into the cell body upon lysosomal biogenesis in the myoid region. And it eventually does make it to the presynapse as well. But as far as mitochondria, it's really interesting. You have all of this in the ellipsoid. So are there fission events occurring from the ellipsoid followed by mitochondrial trafficking along microtubules? And how does it make it to the synapse eventually? So as I mentioned earlier, in order to test our hypothesis, we needed a methodology to disrupt the microtubules. And one of the strategy we took was use of this vinca alkaloid called vinblastine. Now, vinblastine is used heavily in treating leukemia. Okay, it's a cancer treatment drug, and it works by essentially depolymerizing microtubules. So here is a healthy microtubule. You throw vinblastine on, the microtubule starts shrinking from plus end towards the minus end. Okay, this is the mechanism of Finlayson. I'm not going to go into that, but something really cool happens when you treat mice with Finlayson. So these are just intraperitoneal injections for 6 hours. You wait for 6 hours and see what happens to the organelle distribution. There is a complete flip in the lysosomal distribution upon depolymerization of microtubules. So all of the lysosomes that were present in the cell body, they essentially just run back, and all of the new endolysosomes that is being synthesized, they are being synthesized where they are being synthesized, but they have nowhere to go, right? It's just like all of the ships getting stuck in the Strait of Hormuz. There's nowhere to go. So this is where all of the incoming cargo and the lysosomes are stuck now because you don't have microtubules. So this is an indirect evidence to us that maybe there is endocytosis occurring in the photoreceptors. And if we actually quantify again the distribution of mitochondria and lysosomes in these distinct photoreceptor layers, what we find is that unlike controls, the photoreceptor density massively increases in the ellipsoid and the myoid, and it goes away in the photoreceptor cell body, and also pretty much in the synapse. But as far as mitochondria, just doesn't care. Despite waiting for 6 hours and complete microtubule depolymerization in the sensitive, lysosome seems to be much more sensitive to this. This is because what we believe is happening is that the mitochondrial trafficking dynamics is much slower than that of lysosomes. They essentially take these positions very early on, and they don't really move a lot. So just within the experimental time period of 6 hours, we can't do this for longer because this is a category E experiment. It's a heinous drug. The mice don't like it. They get very sick. Um, we we have chosen a dosage well under LC50, so we don't have any fatality in the time period of vinblastine treatment, but it's a very high dosage. And this needs to be kept in mind because there are actual human patients on vinblastine for months, and we don't know anything about their visual function. This is just an image which shows how we do this analysis. We just use image J, and we segment ellipsoid and presynapse based on mitochondrial signal, and we can clearly see mitochondrial distribution, ellipsoid versus myoid versus ONL versus OPL, and this is how the distribution is changing in case of lysosomes. We have done similar analysis, obviously, on controls, n greater than 3 for all conditions, and multiple images in multiple regions have been analyzed. So based on this first round of experiments, what we observe is that endolysosomes accumulate significantly in the inner segment upon microtubule depolymerization, and lysosomal density concurrently decreases in the ONL and the presynapse. But however, surprisingly, what we find is that the mitochondrial distribution really does not change despite microtubule depolymerization in the experimental period. So there are a few things we can conclude. That one is that endolysosome trafficking through the photoreceptor cell body is microtubule dependent, and endocytosis may be occurring in the inner segment. Mitochondrial trafficking is either independent of microtubule organelle trafficking, or the dynamics is much slower than the experimental time period.
I have a quick question before you move on,
Yes.
because I think this is a really amazing result. But I'm also like, okay, you give the mouse vinblastine for 6 hours, and if I'm a lysosome that's kind of in the middle of the outer nuclear layer, where do I go? Because you're seeing almost no signal there. I mean, I'm seeing here
Exactly.
1 or 2. So what happens to the ones that have been stuck, and why are they not still showing up?
So the accumulation that we are seeing in the ellipsoid, we believe, is actually a contribution of two pools of lysosomes. One is all of the lysosomes that were in the cell body But as the microtubules start depolymerizing from the minus end to the plus end, they somehow switch their motor proteins and run back. Okay, they can't just passively get into the ellipsoid. So there is a quick switch of motors that happens, and one of the um, targets that we are interested in is in a nude C. Now that is uh, a machinery that is required for this motor protein switch. And I'm trying to collaborate with Dr. Alicia Gross at University of Alabama, where there is a really beautiful zebrafish model of nude C knockout. And if you knock out nude C, the lysosomes walk towards the plus end no problem, but they can't switch the motors and they get stuck in the synaptic region. Okay. So what we need to do is knock out nude C, hit it with vinblastine, and see if the lysosomes that are stuck at the plus end have nowhere to go and they just get stuck there, right? So that is the experiment we need to do, but that's a really good question.
Yeah, just a question. Yeah, this is an amazing
Yes.
result. Have you tried colchicine?
Sorry?
Have you tried
Yes,
colchicine?
we did not try colchicine. We actually tried cytocalycin D, which is an actin depolymerizing agent. Now, several groups have very effectively used cytocalycin D to depolymerize actin in the photoreceptors, but they use intravitreal injection. What we tried was the easier, dirtier experiment where we did intraperitoneal injections. And unlike vinblastine, the blood retinal barrier is impermeable to cytocalysin D. So if you hit it with cytocalysin D, it doesn't get to the retina. So we did not see any effect for either lysosomes or mitochondria. Or the other possibility, again, is that the mitochondrial trafficking dynamics is um, very slow, so that even if cytocalysin is affecting photoreceptor biology in a matter of 6 hours, we just can't catch it.
Yeah. No, the reason that I'm asking is that a lot of people use colchicine. It's
Okay, we haven't done that. Again, these
that's
are all...
why I was asking. It's for arthritis, for a variety of things. And I was wondering whether there's a particular reason that you did not go for it. You
Yes.
just didn't try. Okay,
No,
thank
we
you.
just did not. We used a different agent and we did not find
Thank you.
any.
Troy, and as your positive control that the drug actually worked is looking at your phalloidin pattern change with the disruption of the active
It
myosin.
doesn't change. We did not see any change in phalloidin labeling.
Well, the
Okay.
vinblastine also, you did see a change, I have to assume.
So there is actually early studies made with vinblastine treatment of the retina. People actually directly injected this heinous drug straight into the vitreous and saw what happened to the photoreceptors. Now, all of the tubulin, microtubule gets depolymerized, but think about the actual amount of protein that has been depolymerized now. It's massive, okay? It cannot be degraded so fast. And all of the lysosomes, they also seem very unhappy when we do this. So how are we going to clear all of this depolymerized protein, either through the proteasomal degradation system or through the lysosomal system, right? It's very challenging. So what people showed during using electron microscopy, this was done in the 60s and 70s, was that you start seeing these crystalline appearance, like crystal-like appearance throughout the photoreceptors, where you just have depolymerized microtubules sitting everywhere. One of the targets that we can try is see how acetylation changes, because with depolymerization, acetylation decreases. That is a target that we are planning on looking at. But clearly, depolymerization has an effect on lysosomal trafficking. All right. So let me go back to this slide here. So how do we remove actins, microtubules? Okay, we got some results pertaining to that using vinblastine. Two, how do you remove actin cytoskeleton? And as I was discussing earlier, we tried cytocalysin D, it did not work, but we still have to somehow disrupt actin functioning in the inner segment and see if endocytosis works. And how do we do that? We really scratched our heads for a few months on this problem, and I suddenly had some synapses just communicating with each other, and it jogged my memory to a paper I had read by Patsy Nishina's group back in 2014, where they showed that the actin cytoskeleton is disrupted in C57 black 6N mice. Okay, so it has the CRUMS1 mutation, which is required for adherence junction formation. And somehow CRUMS1 mutation as a secondary effect causes these ellipsoid zone disruptions and ELM disruptions in C57 black 6N mice. And you can clearly visualize that. You just need to take cross sections of black 6N and you label it with phalloidin, and you see that the phalloidin labeling is discontinuous, and you hit these regions where there is no actin cytoskeleton at all. Okay, so what we did was co-label with LAMP2, and just through preliminary observation, we saw that there were really not a lot of lysosomes in the region of these ELM disruption. So what's happening here? This actually has far-reaching consequence, because these ellipsoid zone disruptions and ELM disruption is also a common disease feature of diabetic retinopathy and AMD. So we are lucky to have access to some of these DR donor eyes in collaboration with Dr. Meg DeAngelis, who just works down the hallway from where my lab is. And here are whole mount images of human retina control, the DR. And if you look at the DR retina, let me just zoom in here, bam, you can see this ellipsoid zone disruptions. Okay, you just lose actin cytoskeleton. There are photoreceptors underneath, the nuclei, everything. You just lose the actin cytoskeleton. Okay, and you can find hundreds and hundreds of these lesions in every diabetic eye, or at least the eyes that I have visualized so far. So, what did I do? There are umpteen number of work that shows that, in fact, ELM and ellipsoid zone is disrupted in diabetic retinopathy, DME, AMD. I've just showed a few papers here, one from Justice Eller's group, one from Michael Abramov's group. And this I found today. This is actually AOSLO imaging of these ellipsoid zone disruptions and ELM disruptions in living, breathing humans. So obviously, these photoreceptors are not happy. And while we all are interested in photoreceptor degeneration, there is a phase of photoreceptor dysfunction that precedes photoreceptor degeneration. So what happens if you lose actin cytoskeleton in the EZ-ELM region, okay? So here is our test. We hypothesized that wherever you lose actin cytoskeleton, where you have these lesions, your lysosome density should go away. These are cross-sectional analysis of just lysosomal density in the inner segment. We just randomly draw squares in the inner segment and look at lysosomal density in each of these squares. And here is what happens in case of black 6J. This is a normal happy C57 black 6J mouse. This is the lysosomal density in C57 black 6J controls. You hit it with vinblastine, the lysosomal density shoots up in the inner segment, and there's a significant increase, right? And we did the same experiment with C57 black 6N, and here is again control C57 black 6N, but we have two squares, okay, green and red, where there is no lesion and where there is lesion, okay? Lesion regions are in red. And we calculated the lysosomal density on cross sections, and lysosomal density in the inner segment is way low compared to controls. And when we hit these mice with vinblastine, complete loss of endolysosomes, almost, in the inner segment region, okay? It's not that we have torn apart anything, it's just a cross section and you have an outer segment. It's just that there is ellipsoid zone disruption and ELM disruption, and you lose endolysosome signal in these regions of regions where actin cytoskeleton is gone, right? So it's really kind of difficult to do this analysis on cross section. So we decided to repeat this experiment and do this on whole mounts. All of these experiments were done in collaboration with Jesse Sloan in the Department of Pediatrics at UB. We again treated Black6J and Black6N mice with vinblastine, and using confocal microscopy, we took 0.5 micron Z size images of the myoid right above the external limiting membrane and measured lysosomal density in Black6J and in Black6N in regions of lesion and non-lesion. What you're seeing down below is essentially the thresholded images for LAMP2. So here is what we find. In case of Black6J, in the inner segment, we clearly see an increase in the endolysosomal content in the inner segment upon vinblastine treatment. In Black6N, in non-lesion area where there is actin cytoskeleton, the lysosomal behavior is comparable to that of Black6J, where you shoot with vinblastine, where there is actin cytoskeleton, endolysosomal density shoots up. But however, when actin is missing, you lose lysosomal density to begin with, and it never recovers, or it does not increase upon treatment with vinblastine, because there really is very few endolysosomes in case of the photoreceptors where there is loss of actin cytoskeleton. So again, this is a very strong indicator that lysosomal biogenesis, endolysosome genesis in these regions. We have also verified this using RAB7, a marker of late endosome. I'm not showing the data here. That endocytosis may be occurring in the inner segment, and it's using actin cytoskeleton. And if you lose that, you just completely lose lysosomal density. And that microtubule obviously affects lysosomal distribution in the photoreceptors. So we wanted to directly test endocytosis in photoreceptors. So what we did was use this was in collaboration with Dr. Shayla Baker at University of Iowa, where we took C57 Black6N mice, and though they were treated with AAV5 EGFP. It was just shot into the subretinal space. We got this nice bleb, and we looked at regions where there was a good transfection. And within an image, I'm just showing phalloidin labeling for actin, and this is the thresholded image for EGFP. And clearly, the EGFP signal in the regions of loss of actin cytoskeleton, EZ disruption, ELM disruption, had very low endocytosis. So this is the average EGFP signal arising from non-lesion area versus lesion area, suggesting that endocytosis is significantly affected in regions of ELM and EZ. So this has multiple implications. The first one is gene therapy even working for CRUMS1, right? People shoot the CRUMS1 gene, just package it in AAV5 or base editing mechanisms, and you just throw it in the subretinal space. But what seems like to be happening is that the sickest photoreceptors are actually not able to take up the therapeutic, okay? So this presents a significant bottleneck in treating essentially any visual disorder that is characterized by EZ disruption and ELM disruption. And two, what happens in DR? Is this the first stage of dysfunction? Because these photoreceptors can't take up anything. No lipoproteins can come in, signaling molecules, right? So how do these photoreceptors survive? We have actually done tunnel analysis, and the nuclei under the lesion area, they're not dead. So these are just zombie photoreceptors that are just not eating anything, okay? So far, we have shown that endocytosis occurs in the inner segment in an actin-dependent manner, followed by subsequent microtubule-dependent trafficking of endolysosomes. Mitochondrial distribution, however, is not affected by microtubule depolymerization, and this has obvious implications in several retinal disorders. So where do we go from here? Okay, so if you lose lysosomes, what happens to organelle quality control in regions of easy ELM disruption? What happens to autophagic flux in these affected photoreceptors? How does mitochondrial turnover occur? How does mitophagy occur in photoreceptors? Can we even really monitor mitophagy? So that is going to be explained by my student Sean, who will be sharing his screen now. Sean?
Thank you.
Let me stop sharing, you share your screen. So Sean has been working on a really interesting project on mitophagy in my lab. Really talented kid, and he has some really interesting insights to share with us today.
Okay, prior to proceeding, we can all see the screen. We can all hear me.
I can hear you.
Yes, Sean.
It would be.
Go to Shears. Yeah, there you go.
Yeah, it's
Go
all,
ahead.
it looks good.
Huh?
All right, thank you. Let me minimize this so I'll be able to actually see where I'm going, and I'll be off. One moment, please. So, because photoreceptors are post-mitotic cells, they rely pretty heavily on maintenance of the organelles that they have and degradation of damaged organelles which may arise. Specifically, the mitochondria is a pretty dangerous organelle because of the fact that it has this propensity to build up reactive oxygen species that can damage either mitochondrial DNA or the phospholipid bilayer that surrounds it. And this can progressively lead to dysfunctional mitochondria, which can either initiate apoptosis or other like cell death pathways. So this is these are mitigated and maintained through different mitochondrial dynamics, right? So we have fission, fusion, and biogenesis, which are the three that are the primary besides degradation, right? So you can have fission of a mitochondria. You have this mitochondria here, which has a bunch of proteins that associate to it. You have DRP-like molecules that constrict around the center or some point on a mitochondria after the contents have been secluded to whatever side for, I believe, unknown reasons. Then fission occurs through, I believe, actin cytoskeleton, further cinching down on that protein and causing two mitochondria, one of which may depolarize, meaning that it loses the proton gradient and has no mitochondrial membrane potential, which would target it for a very specific type of mitophagy called pink-1 Parkin mitophagy. However, if this mitochondria proceeds to be healthy, it is reincorporated into the general mitochondrial reticulum and just stays as it's supposed to be in the cell, healthily functioning. And further, it may, via a mechanism such as mitofusin or other fusion proteins of the mitochondria, like OPA1 for the inner membrane, refuse with another mitochondria to produce larger mitochondria. And these processes are regulated both in regard to the general cell's DNA production as well as the mitochondrial DNA production for biogenesis and also the cell's energy demand. So this can get pretty complicated when it comes to the regulation and such events. Further, we can have smaller fission events that produce mitochondrial-derived vesicles, which are just, again, smaller mitochondria. And these can be targeted directly to the lysosome in a form of like, if anybody's familiar with microautophagy, where the content is shipped directly to the lysosome, it could do similarly and it fuses directly to the lysosome to be degraded that way. Further, we have the concept of mitochondrial exocytosis. I forget exactly which group it was that depicted it, but it was in RGC axons directly at the lamina curbosa at that height that the mitochondria were found to be exocytosed from the cell and taken up by surrounding glial cells, specifically the astrocytes. And then the big question is if there is specific mitophagic function within the photoreceptor cell, which is just the selective degradation of mitochondria, as I will talk more about. So this is really important because dysfunctional mitochondria are also commonly associated with a broad variety of human pathologies. This can include metabolic diseases, neurodegeneration, tumorigenic processes, muscle wasting, etc. So it is very important in general to understand these processes, but also specifically in a very long-lived neuronal system such as the retina to understand specifically these processes. Also, before I go on, feel free to interject at any moment if you have a question.
Quickly describe mitophagy and move to the next slide, Sean.
No, there is mitophagy on the next slide, but my laptop is not
All right,
cooperating.
go ahead. Go ahead.
No, my laptop's not cooperating and allowing it to go. There we are. Okay. So mitophagy is the selective sequestration of the mitochondria or mitochondrial fission blebs, those mitochondrial derived vesicles within an autophagosome, followed by subsequent fusion with the lysosome. It comes in two flavors based on the dependence on the ubiquitin system, right? Ubiquitin proteasome system is also required for the degradation of outer membrane proteins prior to trafficking to the autophagosome or prior to sequestration in the autophagosome and lysosomal degradation. It serves as a primary mechanism for both the degradation of dysfunctional mitochondria and a means for cellular adaptation to energy demands to decrease mitochondrial content. So here we have an autophagosome surrounding a mitochondria uncovered by TEM, transmission electron microscopy, which allows us to see a mitophagy-like event. However, we cannot properly distinguish if this is specifically mitophagy or if this is just a membrane surrounding the mitochondria, because we don't know the fate of this after the fact, right? It's still not going to go. Okay. So, okay. So many chimeric fluorescent proteins have been used and described to differentially target portions of the mitochondria, the outer mitochondrial membrane, the inner mitochondrial membrane, the intermembrane space, all being three separate compartments. And then these are based on the mitochondrial targeting sequence or translocation sequence called the MTS. So these can broadly be described as two categories, including methods for monitoring the mitochondrial fusion-fission dynamics, which include mito-MK2, EGFP, mito-timer, mito-DS-RED, and M-dendra, where M, MT, or mito stand for the mitochondrial aspect of it. These are all separate fluorophores, which have been brought specifically to the mitochondria for these purposes. And then further, there are more novel dual fluorescent reporter systems, as Sri mentioned earlier, such as MitoQC or MitoQC-like variants, such as these two here. And these allow for the monitoring of autophagic processes surrounding the mitochondria, as opposed to just general autophagic processes. There are other further variants, which are crosses between two separate mouse lines, such as the red MIT and GFP LC3B constructs, or MTSRAI, which is a FRET-based sensor. So to just give a rundown on each of these specific models and their limitations, we have mito-MK2 first. It is promoted by a CAG, is promoted by a CAG promoter to express the MK2 far red fluorophore within the mitochondria via a COX-8 mitotranslocation sequence. And while this does a beautiful job of mapping the mitochondria, as shown with co-localization of succinate dehydrogenase, there are a couple of issues. Because of the bleed in to the red channel, excuse me, because of the bleed into the red channel, it is not compatible with other red fluorophores. And also because of the bleed into the far red channel, it is not compatible with other far red fluorophores. So it's kind of a mixed bag when using MK2, and it cannot be crossed with other specific lines used for these research purposes. Further, you cannot properly identify mitophagosomes or mitophagolysosomes simply because of the fact that you are only tracking the mitochondria. You have no construct present to track the phagosome itself or the lysosome itself. While it can be compared with LAMP2, like we had in our previous example, we don't grossly see any level of co-localization between the mitochondrial signal and the lysosomal signal. And then further, we cannot monitor the fusion of a mitophagosome with a lysosome or phagolysosome because there is no acid-sensitive component or other metric by which we can determine if there is a fusion event occurring. Further, we have mito-EGFP, which is similarly promoted by CAG, and then this is transported to the mitochondria via COX-8 MTS again. The primary issue with this model over something like MitoMK2 is the absolute inability to see mitophagolysosomes or mitolysosomes in general, because the green fluorescence, as was mentioned by Shri in the tandem reporter, is the one that is going to quench. So yes, of course.
I
Did
think someone's mic was just on in the background and it captured some sound.
Okay, I thought somebody said question. I'll continue. So as EGFP or GFP species are known to do because of their relatively higher pKa, they quench in an acidic environment. So there is an absolute invisibility of the mitophagolysosome and mitolysosome, because the second that the mitochondria is taken up by the phagosome or the lysosome itself, it will completely disappear from view because of a quenching of that fluorescent protein. So there's no way to track it. Further, again, same issues. You cannot differentiate between the mitophagosome and the mitochondria because there is no tracker for LC3B or other associated proteins to the phagosome, and it is incapable of monitoring mitochondrial fusion with the lysosome or phagolysosome. Now, please work with me. I'm sorry, my computer is not cooperating right now. Okay. So further, there are other mitochondrial tracker models that are not necessarily distinctly for mitochondrial tracking. They serve other purposes, such as DS-RED. This is a mutant of the original DS-RED1. It is E5. This specifically causes a maturation of the protein with oxidation. So it transitions from this green fluorescing state to a red fluorescing state as it matures, but also as it is oxidized. So it allows for a metric of the redox state of the mitochondria itself when it's paired with the COX-8 derived mitochondrial translocation signal. So the issue here, again, falls on the sort of the mitophagosome and mitophagolysosome, as we just simply cannot monitor either of those two structures. There is no way to monitor mitotimer in a sense of mitophagy. There are some publications that insist that fully red puncta, because this is a ratiometric model, meaning that the ratio of red to green indicates the content of oxidized versus reduced, or unhealthy versus healthy in the same respect, mitochondria. And they insist that because the fusion would be an all red puncta, you can determine what is mitophagy versus what is not mitophagy. However, simply because of the fact that you cannot properly identify the membrane structure, and also because you cannot rule out the fact of the redox state, you cannot properly denote whether this is a mitophagic event or if this is some gross oxidative stress inside of the mitochondria at that time point. Further, we have MitoDSRED2, similar to the earlier DSRED1 from the mitotimer. This is driven by a CAG promoter, and then it has it is a mitochondrial translocation signal derived from COX-8, and it is just broadly expressed quite beautifully, in my opinion, in mitochondria of all sorts of cell types, depending on where visualized. So this includes the longitudinal sections of the heart alongside the transverse sections of the heart in a comparison to the wild type. You have liver samples, you have apical microvilli. So it's just expressed everywhere. And this is a true red with a very, very tiny bleed into the far red fluorophore. So again, falls on the sort of the mitophagosome or the mitolysosome. So you can't differentiate between the mitophagosome and the mitochondria because you have no tracker for the phagosome structure itself, nor can you differentiate or monitor rather the mitochondria or mitophagosome and its fusion with the lysosome or the phagolysosome, because we have no acid sensitive or otherwise metric by which to properly track those events. Onto another interesting mitochondrial tracker, we have mitodendrin-2. This is the model itself for the mouse is a CAG-driven expression of a floxed photoswitchable fluorescent protein called dendrin-2. And then again, it is given the NTS by the COX-8 protein. So photoswitchable in this regard refers to the fact that when irradiated with 405 nanometer wavelength light, it is capable of switching from a green fluorescent state to a red fluorescent state. So you can treat a subpopulation of mitochondria with a laser, as we can see down here by this black arrow. There's a red mitochondria, which has been irradiated by that 405 wavelength, and then it slowly comes over, fuses with this fully green mitochondria, and then you have this yellow fluorescent signal, which is the mixing of both the red and the green. So this allows the tracking of the dynamics of the mitochondria, such as fission and fusion, or even biogenesis if you're taking into account total fluorescence within the cell. However, you cannot differentiate, again, between the mitochondria and the mitophagosome. Further,
let's step through fast.
Yep, copy. Further, you're incapable of monitoring the mitochondrial or mitophagosome fusion with the lysosome. And now on to 3 of the very similar variants, which we see with MitoQC. So MitoQC uses, again, that previously mentioned tandem tag, but it uses a Fis1 mitochondrial translocation signal on the carboxy terminus. It's a truncated.
No. Fis1 is the carboxy terminus. It's not the translocation signal per se. It's just the anchor. Okay.
Okay, yes. So it anchors into the mitochondrial membrane via Fis1, and then this allows for monitoring of mitophagic events through this Fis1 protein, or through this tagged Fis1 construct. The interesting thing that is noted with the Fis1 model of MitoQC compared to the other MitoQC models is this very large content of red positive puncta, which we see all throughout the ONL here. These are indicative of mitolysosomes or acidified mitochondrial structures within the ONL, which is indicative of mitophagy and acidification. So this may be indicating the fact that there is aberrant mitochondrial fission with Fis1 expression as the anchor. However, this has not been properly tested, so we cannot say either way. Again, this falls on the sword of the phagolysosome. It does not have the ability to properly monitor the phagolysosome and the mitochondrial targeting to autophagosomes or mitophagosome lysosome fusion events.
The other thing we need to note with this model is that when we performed immunohistochemistry against mito-NKH2 mouse, as I clearly demonstrated, we can only see opposed mitochondria and lysosome, but never prompt mitolysosomes. But it's in complete disagreement with this MitoQC model where they're actually able to observe what is inferred as mitolysosomes. So we can't explain the difference between the two models. Switch to the next few slides.
Just quick question, excuse me. It's not outer segments on the left, it's inner segments, the yellow, right?
Yes, so this is the inner segment.
Yes.
Thank you.
Yes, that
it's
is
just flipped. The orientation is flipped.
Yeah. So again, this is an inducible mitophagy model similar to MitoQC. The only differences are that they use this OMMTS, which as of this morning was identified to be CDGSH, which is an iron sulfur protein domain as a mitochondrial anchor. And they used an inducible mitophagy model. So the construct is under a TET-on control, and it uses an optimized optineurin LC3 interacting region to forcibly initiate mitophagy while inside of the cell when treated with doxycycline. So this one uses a cis1D, whereas this one uses CDGSH. So these have separate mitochondrial translocation signals. But again, this construct is the exact same as the aforementioned MitoQC. And in this entire image, we see maybe 5 punctate red structures prior to the induction of mitophagy in the heart. And this is very, very contrasting with the other reports, which say roughly 200 mitophagosomes per field versus maybe 5. Again,
now let's switch to our model, okay?
Yep, okay. So in further development, we decided to throw our hat in the ring to depict a novel mitophagy autophagy reporter system, which we named Mars. This specifically uses a COX-8 directed MIRFP 670 fluorescent protein to tag the mitochondria and separately tags the phagosomal structure with mCherry EGFP MAP1 LC3B, both promoted separately by CAG promoters. And this allows for separate differentiation of those two components of the cell, which also allows for the differentiation of the mitophagosomes, mitophagolysosomes, autophagolysosomes, autofluorescent puncta, etc. And when paired with a lysotracker, such as a lysotracker blue, this will allow further evaluation. As can be seen from the spectrum on the right, the MIRFP670 is further red than MK2, M-Cherry, and MRFP, and there's very little overlap between M-Cherry and MIRFP670, which allows for these two fluorescent proteins to be used together and specifically distinguished based on maximal excitation, or the wavelength of maximal excitation, rather. And the tandem LC3B tag as a fluorescence reporter construct allows us to discretely determine the stage of the mitophagolysosome within the cell, or the autophagosome within the cell, whether it's independent of mitochondrial content or not. And this construct was specifically knocked in at the ROSA 26 locus to avoid affecting any cellular processes. There are two smaller disadvantages that occur with this model. The pKa of MIRFP670 is at the very lowest extreme of the lysosomal pH itself. So if the lysosome is fully acidified, there is a chance of loss of the mitochondrial signal. However, it is more likely to degrade the mitochondria by that point than to run into issues with the signal. Further, it is a mildly acid sensitive, so it is mildly degradation sensitive, rather, acid hydrolase sensitive, compared to something like MIRFP670 nano, which is a bit more round and has a smaller surface to attack than.
Okay, Sean, let me show the images now, okay?
Yeah.
Okay.
One moment, my computer is not happy in the slightest. Did that stop sharing or no?
No.
One moment. Yeah, it's not. There we are.
Right. Real quick. Okay, so as Sean was mentioning, we have developed this new mouse model called the MARS model, the mitophagy autophagy reporter system. And we have knocked in a mitotracker in the far red region and a tandem tag phagosome reporter in the red and green region, which now allows us to actually visualize all these various compartments: mitochondria, immature phagosome, mature phagosome, mitophagosome, mitophagolysosome, cellular autofluorescence, and when combined with the lysosome immunohistochemistry, about 7 organelles. Okay, so here is our fresh images off the microscope just from yesterday, from last evening. So in the far red region down here is mitochondria, and we are able to visualize a mitophagosome, not a mitophagolysosome, because this is GFP positive for the phagosome, RFP positive for the phagosome, and there is the mitochondria. So we are able to visualize mitophagosome. And in this case, again, we are able to visualize a mitophagosome. A really nice mitophagosome here in the muscle. Here is mitochondria. We can distinguish obviously between immature phagosome and a mature phagosome, but there is a mitophagosome. Okay, so far, people have only been using electron microscopy to detect mitophagy-like events. But what we have been able to do now is develop a mouse model which allows us to faithfully image mitophagy in the mouse. So we intend to use this mouse model to study mitochondrial dynamics and phagolysosomal dynamics of the retina. I don't have images of the retina here yet. And look at the effects of mitophagy upon EZ disruption and ELM disruption. So that's that, friends. That's all we got.
So Sri, you used these two examples, cardiac and striated muscle, is because the mitochondria have such high density, so it's a perfect positive control.
Yes, and also they are tissue where mitophagy, there is example of mitophagy taking place. But what we again notice is that all of these models really differ from the classic mitoQC model. In the mitoQC model, they are overexpressing FIS1, which is required for mitochondrial fission, and overexpression of FIS1 in itself can drive mitophagy. So what we think is happening in the classic mitoQC FIS1 reporter model is that the reporter itself is driving mitophagy, but in all other model systems where they are using RFP GFP tagged to a different mitochondrial outer membrane protein like OMP25, OMMTS, they don't see that. The rate of mitophagy, basal mitophagy is actually very low. And what we are able to do using our model is catch that basal mitophagy process. And while doing that, also differentiate between the transient stages of mitochondria being trafficked to an immature phagosome and that maturing to become mitophagolysosome.
All right. Thank you very much for your talk. Does anybody have any questions?
Just a super quick question. Remind me the background for the mouse.
Black 6J.
Black 6J. Okay.
Yes. What we want to
Thanks.
do is actually cross it onto Black 6N and see what happens to, now if you lose lysosomes, the phagosomes are not going to be maturing in regions of easy disruption. So what happens to mitochondria? First of all, what happens to mitochondrial organization upon easy disruption? And if you lose lysosomes, what happens to autophagy and what happens to mitophagy?
So I'll ask a question, and it's kind of related to the first question that I asked. Why all these CAG promoters? I mean, I know why you would want it, and you can look at cardiac tissue, and that's great. But you know, for a photoreceptor specific phenomenon, presumably Mueller cells have, you know, all these same things. Wouldn't it be nice to have markers that are somewhat cell type specific? And then if you want a very general Cree system, you can sort of use this as a LOX-P Cree and breed it to a general Cree.
Actually, that's a great question. We didn't have enough time to go over all the mitotracker models. There is a new model called the kaleidoscope model, which is cell type specific knock-in, but still it's using CAG. So to use cell type specific promoter, it becomes a really expensive model to develop because when we developed this model, we just have kept essentially the all fields in mind. Anybody interested in the brain, exercise physiology, anybody can be able to use the mouse model. But to be able to study the process in a cell type specific manner, yes, there is a conditional knock-in model called um, the kaleidoscope model. But again, it uses a CAG promoter, mostly because it almost guarantees expression, but it comes with a problem, as we demonstrated in this paper, because you're driving LC3B expression using CAG, this construct is being overexpressed several folds over the endogenous levels of LC3. And when people do microscopy, because EGFP quenches really fast upon lysosome fusion, you really don't see any phagosomes at all. Okay. In case of photoreceptors, 99% of the phagosomes are mature phagosomes. There are very few immature phagosomes that you can capture using this model. But we have seen and actually reported several cases of just artifacts because of using CAG. People are overexpressing this cytosolic protein. And if you just increase the gain high enough on your microscope, the entire photoreceptor lights up with just GFP and RFP. And several studies, even published in high impact factor journals, have actually misunderstood and misinterpreted what this increased cytosolic LC3 signal means in these CAG-driven models. I completely agree. CAG is a problem, but it's currently the best solution we have.
All right, does anybody else?
Even if you run it using Rho-I-Cree, rhodopsin is expressed at a very high level in photoreceptor, and LC3 expression is nowhere comparable to rhodopsin. So even if you use a cell type specific promoter, you could still be significantly overexpressing the construct compared to the endogenous levels.
Yeah, I mean, totally agree. You can always, of course, check if with the appropriate controls if you're influencing, tissue health by overexpressing a particular construct. So does anybody else have any final questions before we go? And if not, thank you both for wonderful talks. And I guess I'll see you all in a few weeks.
Thank you so much.